Hello everybody, welcome to the fourth session of chapter Human Health and Diseases. In our last class, we have come across different types of immunity, that is active immunity, passive immunity. We have learnt how the immune cells, or either it may be inherited or it can be artificially transferred into our body. We have come across allergy, what are the causes of allergy, how it can be prevented or cured. We have come across autoimmune disease. Now in today's class, we will start with immune system in our body or immune organs which are present in our body. So immune organs or immune system includes lymphoid organs, tissues, cells and soluble molecules are proteins, especially the antibodies. So all these come under immune system, that is lymphoid organ, tissues, cells, soluble molecules, antibodies are the examples for soluble molecules. When it comes to lymphoid organs, there are two different types of lymphoid organs. One is primary, another is secondary. Now primary lymphoid organs include, that will be bone marrow as well as thymus. And when it comes to secondary lymphoid organs, it includes spleen, Peyer's patches, of small intestine, lymph nodes, appendix and tonsils. So these are all the secondary lymphoid organs. What is this lymphoid organ doing, or what is the function of the lymphoid organ if you ask? Lymphoid organ helps in origin, maturation and development of particular types of cells. Those cells are called lymphocytes. So lymphoid organ helps in maturation, especially the origin, maturation and development of this lymphocytes. All the three components what I have told, that is origin, development as well as maturation, everything takes place in the primary and secondary lymphoid organ. Now when it comes to bone marrow and thymus, in this the lymphocytes, normally these get originated and they undergo development to become antigen sensitive. So here in the primary lymphoid organ, what happens is, both in the bone marrow as well as the thymus, the lymphocytes develop, undergo maturation and they become antigen sensitive lymphocytes. What does antigen sensitive means? The ability of the lymphocyte to trap this antigen and to destroy them. That maturity will be developed in primary lymphoid organ. Once the lymphocytes are matured in the primary lymphoid organ, next they get transferred into the secondary lymphoid organ. In the secondary lymphoid organ, there will be spleen, Peyer's patches of small intestine, lymph nodes, appendix as well as tonsils. So here in the secondary secondary lymphoid organ, the matured lymphocyte undergoes proliferation. So now they have become antigen sensitive in the primary lymphoid organ, whereas in the secondary lymphoid organ they undergo themselves division. They increase their number, that is what is called proliferation. So proliferation process takes place in the secondary lymphoid organ and then they become effector cells. So in the secondary lymphoid organ, these undergo proliferation. These I mean the matured antigen sensitive lymphocyte which have been transferred from the primary lymphoid organ. They undergo proliferation, they increase their number and become effector cells. This is how the lymphocytes within the lymphoid organ, that is the primary and secondary lymphoid organ, produce, or they get produced like origin, they undergo development, they undergo maturation. So here lymphoid organs, the lymphocyte special type of cells which are produced are termed lymphocytes. Primary lymphoid organs include bone marrow and thymus, where the development of the lymphocytes takes place and they become antigen sensitive. Once they become antigen sensitive, they undergo maturation and reach the secondary lymphoid organ. In the secondary lymphoid organ, they undergo proliferation, that is they increase their number. Once they increase their number, they become effector cells. Now we will be studying in detail about some of the primary as well as secondary lymphoid organ. To study in detail, first we will concentrate on the primary lymphoid organ. That is the bone marrow. Bone marrow is the particular or primary lymphoid organ. It is the region where the blood cells as well as the lymphocytes are produced. So bone marrow is the region for origin of the lymphocytes. Not only lymphocytes, even almost all the types of blood cells are produced in the bone marrow. Next is thymus. So location of thymus, it is in the chest cavity, near the heart, below the breastbone. So by birth, so thymus will be present in larger size during birth. As you develop or as the human being develop or grow and reach the puberty, the thymus will start to reduce its size and become very small. As you have noted that old people have lesser immunity, that is because the thymus will be very much reduced in them. So as you get older, thymus gets reduced as well as your immunity. This is a natural process which takes place in our body. So during birth, the thymus size will be very big. As you reach the puberty, what happens? The size of the thymus will start to reduce. So the location of thymus, it is present in the chest cavity, nearer the heart, below the breastbone. What is the function of this bone marrow and thymus? Both bone marrow and thymus, these provide microenvironment. Microenvironment will be provided by the bone marrow as well as the thymus, so that they will become antigen. They, in the sense, lymphocytes become antigen sensitive. So the microenvironment provided by the bone marrow and thymus contains certain types of antigens which the lymphocytes will be focusing on, or they will be learning about this particular antigen, identifying it and trying to produce antibodies against this antigen. So this microenvironment will be provided by the bone marrow as well as the thymus. So this comes under primary lymphoid organ. Now next one is secondary lymphoid organ. First we will study about spleen. So spleen, it is a bean shaped organ. So in this spleen, what is the main function or what does it do is, spleen helps in, or it is the reservoir of It is the reservoir or the storehouse of RBCs or erythrocytes. They help in storing the RBCs as well as erythrocytes. At the same time, the matured antigen sensitive lymphocytes enter into the spleen. So now while they enter, what is the function of spleen is, they help in trapping antigen or microbe organisms. The antigen as well as microorganisms are trapped, especially the blood-borne antigen and microorganisms are trapped by the spleen. That is the main function apart from storing RBCs or erythrocyte is done by spleen. They help in trapping the blood-borne, ರಕ್ತದಲ್ಲೇ ಇರುವಂತಹ blood-borne antigen as well as microorganisms. Once they trap this, what will be done? These antigen which are trapped by the spleen are used to activate the lymphocytes. So the spleen traps the microorganisms as well as antigen which is present in the blood. Once the antigens or microorganisms are trapped, they are used in activating the lymphocytes. So these antigen are used as training system where the lymphocytes will identify the microorganisms or antigen, tries to provide antibodies against these microorganisms or antigens. So that is how spleen works, by trapping the blood-borne antigen or microorganisms. That is about secondary lymphoid organ, spleen. Next secondary lymphoid organ will be lymph node. Lymph nodes. So these lymph nodes are dot-like structure or node-like structure which are spread throughout the lymphatic system. They also have the same property just like spleen. These help in trapping the microorganisms or antigens. These microorganisms or antigens which are trapped by lymph nodes, all these try to enter the fluid, that is the tissue fluid or body fluid of our body. So that is why these lymph nodes trap these antigens as well as microorganisms in the body. So that is about the lymph nodes. So if you see the location of the primary and secondary lymphoid organs in our body, if you consider that this is human body. So if you see here, this is the chest region, here will be the thymus that will be present in the chest cavity, and when you see here in the hand region or arm region, you can find the lymph nodes where the lymphatic vessels will be present in the thigh region, that is in the lower part of the body, that is what we call lymphatic system. So this is the location of different forms of lymphoid organs present in our body, both primary as well as secondary. Next, apart from this one, 50% of the lymphatic system constitute of this main lymphoid organ, that is malt. Now malt means mucosa associated lymphoid tissue. So this is mucosa associated lymphoid tissue, that is malt. Where is the location of this malt, if you ask? Malt is located in the inner lining, that is this mucosal lining of the different tracts like reproductive tract, respiratory tract, urinogenital tract, digestive tract, etc. You can find this malt which helps in again trapping the antigens or microbes. Almost 50% of the lymphatic system consists of this malt which is present in the innermost lining of the systems or the tracts like digestive, respiratory as well as reproductive or urinogenital tracts. So that is about different types of immune system present in our body. Next we'll concentrate on one of the infectious disease, that is AIDS. To define, this is not a single disease. To elaborate this one, acquired immunodeficiency syndrome. So, acquired immunodeficiency syndrome, here it means that it is not a single disease. Initially what happens is our immune system is suppressed or reduced, and because of this immune deficiency it leads to syndrome. Syndrome means it is group of different types of diseases affecting our body. So that is what is AIDS, acquired immunodeficiency syndrome. So it was initially identified in 1981, but now more than 25 million have this AIDS. So to know properly about this AIDS, we should know initially that how does it transmit. I have already told you this is an transferring, that is transmitting type of disease or infectious disease. So here we should know the reason how do they get transmitted. The transmission is mainly because of sexual contact with infected person. So this sexual contact with infected person is mainly if it is having only individual or one partner, the chances of having AIDS is very rare, but if it involves multiple partners, then the transmission of AIDS is at very high risk. That is one of the reason of transmission, having a sexual contact with multiple partners. Second one is through using common syringes, needles. These use of common syringes or needles, especially with intravenous or those who use, or we call drug abusers, who use common syringes or needles to insert the drug inside their body, that is intravenous drug which is transferred into their veins. So that is why the chances of having this AIDS is at very high risk, using common syringes as well as needles. Next one is through blood transfusion. So blood transfusion, improper ignorance regarding the blood transfusion, that is blood when you donate the blood, when somebody receives the blood from the blood bank, if at all the individual is having AIDS, then the chance of spreading this AIDS will be at very high risk. That is sharing or transferring or donating the blood, that is blood from an infected person. That is blood transfusion helps the increased risk of transmitting this AIDS. And next one is through that is from an infected mother to a child. So there are chances that from infected mother to child this symptoms will be, or the AIDS will be transmitted. If the mother is infected by AIDS, then the child also will be at a high risk of, through the placenta, the child will also be at a very high risk of having AIDS. But nowadays there have been certain medical processes through which we can protect the child even though the mother is infected. So these are the 4 different ways of transmitting this acquired immunodeficiency syndrome. So these are the 4 ways of transmission. Now we should know what is the one which causes this AIDS. So the AIDS syndrome, it is caused because of HIV, that is human immunodeficiency virus, that is HIV, human immunodeficiency virus. This human immunodeficiency virus belongs to a family, that is retrovirus. So how the retrovirus will be is, retrovirus will be having a protein coat or a envelope, and inner side it contains the genetic material RNA. So the RNA genetic material covered with the protein coat belongs to the retrovirus family, where HIV is one of the example. Now we have to study how this HIV virus, we know the way of how it gets transmitted into the body. Now we have to know the second step, how this virus reduces our immunity or makes our body immunodeficient. For that purpose, you should know how the HIV virus replicates within the body. So we will be studying the diagram of replication of HIV virus. Okay, so this is the diagram regarding how there is replication of HIV virus or the retrovirus. Now initially the virus, once it enters into the body through the different 4 ways what I have explained regarding the transmission process, once they enter into the body, so the viral, as I have told, HIV virus is a retrovirus, so it is having a protein coat outside and the genetic material will be virus RNA. This identifies or enters into a particular cell, that cell will be macrophages. So these macrophages are the cells which the HIV virus first or initially enters into. The macrophages enters inside, the whole virus doesn't enter into the macrophages, only the genetic material will be entering inside the macrophages, leaving the protein coat outside. Once the viral RNA enters into the macrophages, the viral RNA undergoes a process called reverse transcription. So the process is here reverse transcription. Because of reverse transcription, the viral RNA gets converted into viral DNA with the help of an enzyme called reverse transcriptase. So the process here is reverse transcription, because of which one the viral RNA gets converted into viral DNA with the help of an enzyme called reverse transcriptase enzyme. Once the reverse transcription process finishes, the viral DNA will be present within the host cell. This viral DNA incorporates into the host genome. The nucleic acid of the host will be incorporated with this viral DNA. Once the viral DNA incorporates with the genome, the host genome will be made in such a way that they start producing the viral RNA. So initially it was virus RNA entering inside, undergoing reverse transcription process to produce viral DNA. Viral DNA is produced because the nucleic acid in the animal cell will be DNA. So to incorporate this viral DNA, it requires DNA, both should be DNA, or else if the RNA is there, the incorporation is impossible. So viral DNA incorporates with the host genome, then they start dividing or they start producing viral RNA. So by the end of this genome process, once the incorporation takes place, the cells or the host cell will be producing viral RNA. So viral RNA, as they produce, the protein coats will start to surround themselves, and the viral DNAs get converted into RNA, and the new virus will be released from macrophages. As macrophages produces large, very much number of viruses, that is why macrophages can be termed, it is called HIV factory, as they produce large number of viruses. When the cell, when the host cell or the macrophages, when they are undergoing the production of virus, dividing of virus as well as release of this virus, the host cell will be alive. This macrophage or the host cell will be present when they undergo this division or production of smaller cells. Once the virus is produced or released from the macrophages, they affect the T helper cells, that is the TH cells or TH lymphocytes. Thymus helper lymphocytes will be affected, which indirectly affect the development of T lymphocytes or T cells. So once they affect the T cells, automatically the immunity will start to reduce. Immunities will start to reduce means the individual will develop bowel, diarrhea, fatigue, loss of appetite within almost 5 to 10 years. That means AIDS, it takes almost 5 to 10 years to exhibit its symptoms. So it will be almost about 5 to 10 years. Once the symptoms will start coming, the body T cells will always starts to reduce. Once T cells start to reduce, automatically immunity will also be reduced. That means our body will be a open area for bacteria, viruses, fungi, parasites to attack. bacteria, especially mycobacteriums, are the one which affect our body. Apart from these mycobacteriums, there are also parasites like toxoplasma. So my bacteria like mycobacterium, and when it comes to parasite, it will be toxoplasma. These are the important bacteria and parasites which affect our body if the individual is having AIDS. If not AIDS, then these mycobacterium and toxoplasm can be easily removed from our body with our healthy immune system. As this retrovirus affects the macrophages and then the T helper cells and finally the T lymphocytes, it affects the immunity. That is why our body will be an open part or open place, so that the bacteria, virus and fungi can easily affect our body and cause different varieties of disease. So here there is actually no cure for AIDS, but still prevention is present. So prevention means the way with which transmission takes place, all those should be avoided or it should be done under proper guidance. So to identify if the organism has AIDS, ELISA is the test which can be done. So what is ELISA? Enzyme linked immunosorbent assay. This is the test by which we can identify whether the individual is having AIDS or not. So that is about different ways how the AIDS can be transmitted or how the HIV virus or the retrovirus undergoes replication within the body. So a replication within the body initiates by viral RNA entering into the body, into the host cell, especially they affect the macrophages. The viral RNA undergoes reverse transcription to produce viral DNA, which incorporates with the host and they divide to form or they produce viral RNAs. These individual viral RNAs will develop a protein code and will be released into the body. Once they are released into the body, they first affect the T helper cells, that is the thymus helper lymphocyte cells. Once they are affected, automatically their next target will be T lymphocytes. Once the T lymphocytes will be affected by this virus, the automatically immune cells or immunity within our body will start to reduce. Once the immunity is reduced, the body is an open plate for different kinds of viruses, bacteria, fungi, etc. or parasites to attack our body, where a particular bacteria called mycobacterium will be the first one to affect our body or adversely affects our body, along with the parasite which belongs to toxoplasma. So that is how our body will be affected by different types or of different forms of diseases, where the multiple symptoms can be observed like bowel, fever, appetite, loss of appetite. It also even leads to diarrhea, constipation, etc. So all these shows or all these symptoms can be seen from 5 to 10 years, within 5 to 10 years, as it is very long process, it takes a very long time to show the process. So that is about AIDS as well as how it is caused. In next class, we will be studying about how prevention methods can be taken for this syndrome, that is acquired immunodeficiency syndrome, that is AIDS. So in today's class, we have learnt different forms of lymphoid organs present in human body, as well as we have come across the causes as well as the transmission, as well as how the replication takes place within our body for acquired immunodeficiency syndrome. Thank you.